Biochemistry for microbiology. A living cell is a complex system which consists of many chemicals. Our body cells or any cell, it utilizes these chemicals to obtain energy and synthesize cellular structures. Thus, an understanding of chemistry is essential to an understanding of living organisms. Study of these chemical reactions which are going on in the cell, it is called as biochemistry. It is a branch of chemistry. We'll be learning about few concepts from chemistry which are essential to understand this particular branch. What are chemical reactions? The interaction of molecules, atoms, or ions resulting in the formation of one or more substances is called as chemical reaction. So it involves breaking and formation of chemical bonds. Bonds means linkages between atoms. Every living organism is capable of carrying out many chemical reactions to stay viable or to stay alive, to grow and to reproduce. Microorganisms perform a great variety of chemical reactions. Some result in conversion of nutrients to cellular substances, where simple compounds become large, complex molecules. Some chemical reactions result in the breakdown of large molecules into smaller molecules or atoms, all put together, whether it is degradation or biosynthesis. The total of these biochemical reactions is called as metabolism. So, biochemical reactions which occur inside a cell, whether they are degradation type of reactions or biosynthesis type of reactions, are called as metabolism. So we can say metabolism as a sum of anabolism and catabolism. Catabolism, it involves degradation of chemicals to obtain energy. We can say it is the degradation of biomolecules to obtain energy. It involves breaking of bonds. Anabolism involves biosynthesis of the cellular components, the components of cell. It involves formation of bonds and it involves biosynthesis of biomolecules. In these types of reactions, the energy liberated is utilized. So, catabolism involves release of energy and anabolism involves consumption of that energy. All the chemical reactions are reversible. Some chemical reactions involve a single compound that undergoes some change in its molecular structure, resulting in the formation of a new compound. For example, compound A is converted to compound B. We can see the arrows are in forward direction as well as in reverse direction. This means this reaction is reversible. Now, anabolic reactions. Anabolic reactions involves synthesis of new molecules by the formation of new bonds between two or more molecules, ions or atoms. The energy is required for the formation of these bonds. So energy is consumed during anabolic reactions. You can see this reaction that A, it may be an atom, ion or molecule, combines with B, which may be atom, ion, or molecule, it leads to synthesis of a new compound or a new molecule. AB. This is anabolic reaction. In living cells, what are the different examples of anabolic reactions? The examples are the combination of amino acids to form proteins. That means amino acids come together to form protein. The combination of sugar molecules like monosaccharides. Monosaccharides combine together to form polysaccharide, for example, starch. In case of formation of nucleic acid, nucleotides combine together to form nucleic acids such as RNA and DNA. Glycerol molecules, when combined with fatty acids, leads to formation of triacylglycerol, which is fat. Triacylglycerols are all these are the examples of anabolic type of reactions which involves biosynthesis of biomolecules like proteins, fats, carbohydrates and nucleic acids. Catabolic reactions. Catabolic reactions involve degradation of large molecules into smaller units such as ions, atoms, or molecules by the breakdown of chemical bonds present between them. So, during breaking of bonds, the energy is released. So, energy released during these reactions, it is used in anabolism. So, it is a type of cycle. As an example, AB is a molecule is broken down to A plus B. During this process, the energy is released. In living cells, the examples of catabolic reactions are degradation of fats to fatty acids, in which the energy is released in the form of ATP. Proteins are degraded to amino acids. Polysaccharides are degraded to monosaccharides, followed by their further degradation. So all these are the examples of anabolic reactions and catabolic reactions. Next, all biochemical reactions which occur in the cell are mediated or catalyzed by biological molecules called as enzymes. Enzymes are the organic biological catalysts present in every cell. They are present in all the living organisms, including viruses. Catalysts are the substances that, even in small amount, increase the rate of chemical reaction. That is the definition of a catalyst. A catalyst is not destroyed or consumed during a chemical reaction. That is the property of catalyst. Enzymes, they differ from inorganic catalysts because they catalyze a specific type of chemical reaction. That means enzymes are highly specific for the reaction that they carry out. All enzymes are proteinic in nature except RNA splitting enzyme, which is called as ribozyme. We need to remember this. Ribozyme is an example of an exceptional enzyme which is made up with nucleotide. It is also a RNA splitting enzyme. These are the general properties of enzyme. Again, enzymes are beyond their specificity for the substrate. Enzymes are highly sensitive to environmental parameters like pH and temperature, metal ion concentration, and the surrounding chemical and physical environment. This is enzyme catalyzed reaction, in which we can see enzyme binds to substrate, so which is shown here as enzyme plus substrate. Then after binding, there is formation of a complex called as ES complex, that is enzyme substrate complex. Further, this enzyme substrate complex further gets converted to or developed into enzyme product complex. Here you can see that the substrate is now converted to product by this enzyme. So it is called as enzyme product complex. Then after that, after the reaction is complete, after the product formation, enzymes then separate from product. Again, this enzyme can go back to interact with a new similar or identical substrate, new molecule of substrate, and again this reaction proceeds. So at the end of every reaction, there is this result enzyme plus product, and again this enzyme is ready to uptake new substrate. So this is how a single enzyme can be used multiple times for the formation of the product. This is enzyme molecule and this is substrate. What do you see here? That this diagram shows the specificity of an enzyme for the substrate. So this is just an indication that shows the specificity. So a substrates and enzyme specificity. So this is the site where the substrates bind. This is called as active site. So active site is the position on an enzyme for the binding of appropriate substrate molecule. Then mechanism of enzyme activity as it was shown in this reaction. We'll see this in the form of a diagram. So this is the mechanism of enzyme activity. You can see that this is the substrate having combination of molecules. Say for example, this is the enzyme which is specific for this substrate. So it binds to that particular active site. After that, there is a transition state. Transition state is characterized by the formation of enzyme substrate complex, and followed by that this substrate is converted to product. In the next reaction you can see that this product, initially it was an intact molecule made up with a combination of molecules, and now at the end this is the product that the molecule is broken down into two different products. This is a cleavage type of reaction catalyzed by enzyme. Means the substrates are broken down to different products. This is an example of enzyme catalyzed reaction. Another example that enzyme of enzyme catalase reaction is this. Here we can see substrates are two different molecules. They have an active site on this enzyme. So these substrates then bind to this enzyme's active site. Then there is a transition state, that is enzyme substrate complex, and you can see here that these two molecules are now combined together to form a single product, in contrast to earlier slide. Here there is biosynthesis, and in the earlier slide it is degradation. So enzymes are responsible for the participation in cleavage types of reactions, that is breakdown type of reactions, as well as biosynthetic type of reaction as shown in this slide. After the reaction occurs, the enzyme is released from the chemical reaction and it combines with another substrate. So this reaction is repeated many times until the reaction reaches to equilibrium. So it is a state in which both the reactants and products, the concentrations are balanced. One enzyme molecule can catalyze the conversion of many substrates to product, and reactions are millions times faster than same reactions without enzymes. The important chemicals present in living organisms are carbohydrates, proteins, lipids, and nucleic acids. Therefore, like all matter, living organisms contain atoms and molecules as their most basic structural unit. Examples of the enzymes. We can see this table. The first column states the substrate, second column states the enzyme, name of the enzyme, and the third column states the name of the product. So if you see the first row, the first column, if substrate is lactose, and the enzyme is lactase enzyme, and this lactase enzyme, when it acts on lactose, it leads to formation of glucose plus galactose. That means it is a degradation type of reaction catalyzed by this this lactase enzyme. Another example, it is substrate is maltose, enzyme is maltase, and it is responsible for the formation of glucose. So maltose is a disaccharide, it is broken down to 2 units of glucose. So again this is a cleavage type of reaction. Cellulose is the substrate, and the enzyme cellulase, when it acts on cellulose, leads to formation of many units of glucose, because cellulose is a polymer which is made up with monomers of glucose. So again this is a cleavage type of reaction. Next example is lipase. Lipase enzyme, the substrate for lipase enzyme is lipid. It acts on this polymer, it leads to the formation of glycerol plus fatty acids. Again it is a cleavage type of reaction. Amylase enzyme, this enzyme acts on the substrate called as starch, which is a polymer, and it is broken down to glucose and maltose. So again this is a cleavage type of reaction. Proteases are the enzymes which act on the substrate protein, that leads to formation of peptides and polypeptides. So in this way, these are the examples of different enzymes. One thing you might have noticed, that as it is darkened, you can see the color influx here, ASE. So if you see the names of all the enzymes, the names ends with ASE. This means that This is a suffix. uh, Due to this suffix we can identify the name of enzyme. This nomenclature system of this enzyme, this is one of the key factor for nomenclature system of the enzyme, that there is use of this suffix, that is ase. Another nomenclature criterion is there may be inclusion of the name of the substrate, lactase. So as the name indicates, it indicates the name of the substrate is lactose, maltase maltose, cellulase cellulose, lipase lipid, amylase starch, protease protein. This is how the nomenclature system of enzyme works.